Hi, welcome to the second part for our video lecture in the topic genetic engineering. So last time we talked about reproduction, the central dogma of molecular biology, where we explored three different processes: the DNA replication, transcription, and translation. So if you think of it, all those processes are essential in the survival of living organisms. And with this knowledge, it opened a lot of doors for scientists, and the field genetic engineering was born. Okay, so for today, I want to start with a question. So, what are genetically modified organisms, or what we call GMOs? I think you are quite familiar with this. Maybe you have encountered this word before. So, you can pause the video right here and gather your thoughts on it. Okay, and if you're done, I want to show you a video.
Strange genetically modified products. Vaccinated bananas. Researchers are now experimenting with injecting vaccines for hepatitis B and cholera into banana saplings. As the plant grows, the cells produce the virus proteins, but not the infectious part. So in the future, all people need to do to be protected against disease is bite into a banana. Researchers have also done similar experiments with carrots, potatoes, lettuce, and even tobacco. Purple tomatoes. The Johns Innes Center in the United Kingdom has been experimenting with adding anthocyanine to tomatoes and ends up giving them a darker color. Anthocyanine can be found in grapes and blueberries and gives them the darker skin, which is full of antioxidants and helps prevent cancer. But it will still be a while before blomatoes make it on the market. Frankenswine. Farmers have been dealing with an onslaught of toxic poop from their pigs due to the food they're feeding them. Pig feed in the US and Canada generally consists of corn and cereal grains, but the result is phosphorus-laden poop. Farmers tried putting phytase to help their pigs digest the phosphorus with less than successful results. So Canadian scientists have created a genetically modified pig called Canadian EnviroPigs, who are bred to make their own phytase and therefore have less toxic poop. Venomous cabbage. Scientists in China have managed to crossbreed the cabbage and scorpion genes together to create a cabbage with its own venom in order to reduce the amount of pesticides used. So when caterpillars and other bugs bite into the cabbage, they die instantly. But what about humans? Apparently the cabbage is modified so that humans aren't affected by the poison. Square watermelons. Square watermelons have become the center of a hot debate. The trend of these shapely melons have taken off in countries such as Japan. On one end of the argument, researchers have claimed responsibility for the genetically modified product and claim that the watermelon isn't edible and more for decoration. On the other end, farmers claim that you can create your own edible square watermelon by growing one inside a strong box. Glowfish. Take a natural zebrafish and combine its DNA with bioluminescent jellyfish, and you have your first genetically modified animal that is marketed as a pet. Glowfish was originally created to be a warning sign for pollution, but researchers and scientists realized that if you add a few extra colors, you have a market in the pet industry. Humanized cow milk. In China, scientists introduced the human genome to bovine embryos in 2011 so that they can breed cows to produce humanized milk. Basically, this means that there are now cows that can generate milk that is identical to human breast milk. The experiment has been rough, as 10 of the 42 cows died upon birth and several more since then. So the idea is great, but the execution is still being worked on. Flavor Saver tomato. The Flavor Saver tomato is the first one to be grown genetically and be cleared for human consumption. A California-based company called Calgene added an antisense gene to the tomato, ensuring that the ripening and rotting of the tomato is slowed down. But the idea hasn't quite taken off despite being in the works since 1994. First, the tomatoes were too delicate for transportation, and then they were too bland for consumption. Pollution plants. The University of Washington is working on genetically modifying plants that will help fight pollution. The idea behind the experiment is that the plant will absorb the chemicals and pollution in the groundwater and break down the pollutants and release them into the air. In these newer plants, they remove about 91% of contaminants, where regular plants only remove 3%. How do you feel about some of these strange genetically modified products? Let us know down below. Thanks for watching.
All right, there you go. So that was a video from the YouTube channel The Richest. And it showed us a lot of genetically modified organisms. Okay, so in genetic engineering, it involves the use of plants and animals with desirable traits to reproduce organisms with a specific trait. And this is also called selective breeding. So, for example, in the video, they gave us the example of the flavor saver tomato. And the tomato had desirable traits on it, which made it long lasting. So the rotting and the ripening of tomatoes is slowed down. So those are the desirable traits that people wanted the tomato to have. Also, crossing of plants or animals with different variations of the same trait to produce an organism with the best trait, which is also known as hybridization. So genetic engineering opened the doors for hybridization. And just an example, a mule is an example of a hybrid animal. It's a best known example. It is an offspring of a female horse or mare and a male donkey or jack. And mules are widely used as weight bearers. So that's an example of a hybrid. Okay. Moving on, genetic engineering has allowed a specific gene from one organism to be transferred into the DNA of another organism. So the knowledge for DNA will also be applied here in genetic engineering. Okay, let's take a look at this bacteria that I have here, the E. coli or the Escherichia coli. And this bacteria played a very important role in genetic engineering because scientists were able to use this bacteria as a medium for different processes in genetic engineering, which I'll be able to show you later. Okay, in recombinant DNA technology, this is the technique of combining two DNA sequences from different sources. One goal of this technique is cloning. Are you familiar with cloning? Okay, do you know Dolly, the sheep? So cloning a particular gene for analysis or mass production of a medically useful protein product such as insulin, which is an important hormone to regulate blood sugar levels in the body, and the human growth hormone as well. Okay, so the field of genetic engineering opened a new market for medicine to help people with different conditions. Okay, so I'll show you an example. Let's zoom in into this picture over here. So here we can see a human cell, and inside this human cell we can find the nucleus, which contains our DNA. And here a gene is taken for human growth hormone. So this specific gene codes for the human growth hormone. And then another organism is taken, particularly a bacterial cell. So they chose a bacteria, and the bacteria has chromosomes in it, and it also has a plasmid. So a plasmid is also a chromosome, but it's a circular chromosome. So this plasmid is taken from that bacterial cell, and it will be cut open, and then our gene from the human cell earlier will be combined. So DNA recombination will take place. After that, we now have our recombinant DNA. So this means that this is a bacterial plasmid which contains the gene for human growth hormone as well. This recombinant DNA here will be inserted into another bacteria. So you see, the DNA insertion will take place. We now have the bacterial cell for containing gene for human growth hormone. Ooh, so this is what happens. This is what it's like for the recombinant DNA technology. Okay, so I'll show you the steps in producing these transgenic organisms or the genetically modified organisms. Okay, but you might be curious, why are they using bacteria? Out of all the organisms in this planet, why did scientists choose to use bacteria? Okay, so it is like that because these bacteria, if you think about it, they reproduce through binary fission and they also reproduce exponentially. So it's in the form of asexual reproduction, very efficient and also very fast. And they multiply exponentially. So in a short span of time, and they can obtain a lot of samples. Okay, so let's get onto the steps. So the first is to obtain DNA from a source cell. So let's zoom in. Here we have our human cell, right? So this is the chromosomes inside the human cell. And we need to obtain the DNA from it. The human cell is our source cell. Okay. So the chromosome will be sliced up and the gene will be isolated. The gene here in this example is responsible for controlling formation of insulin. Okay, insulin. And then the next step is to insert DNA into a plasmid. So this requires us to look at this side, the other side of the picture. So we have our bacterium here. This bacterium contains chromosomes just like the human cell. So it's labeled here in red. And we also have the plasmid. So the plasmid is another DNA containing structure inside the bacterium, but it's different from the main chromosome of the bacterium. So this plasmid is isolated and it will be cut open using enzymes. Okay. And then the third step is to construct a recombinant plasmid. So we need now the gene from the human cell to be combined with the plasmid from the bacterium. Okay, so after that, gene inserted in plasmid. So we now have our recombinant plasmid. Up next is to insert the recombinant plasmid into the recipient cell. Okay, so we have a recipient cell. It requires another organism. So we have a suitable bacterium over here. Okay, and this bacterium, this is where the recombinant DNA will be inserted. So you see, we now have this one, the plasmid with the yellow color. The yellow part, this is the gene for insulin. Okay, and then this suitable bacterium will reproduce through binary fission. And this will now result in the production of insulin. Okay. So that's how they do it. With this, insulin will now be created, which will be available to people with different conditions, which need to have insulin in their bodies. All right. Okay, so I just showed you some steps of how transgenic organisms are created and how they work. So what do you think? Earlier I asked you if you know what genetically modified organisms are. So now I want you to think, is genetic engineering boon or bane? If we say boon, then we think it's advantageous. And if we say bane, it's disadvantageous. So what are your views about this? We can pause the video here. Okay, let's proceed. So, I will show you some applications of genetic engineering. First off, in pharmaceuticals. So, just like our previous examples, genetic engineering will have applications in pharmaceuticals such as insulin, human growth hormones, also in blood clotting factors and fertility hormones. So, it helps people with different conditions. It opens a new market for them so they can avail these different products, medicinal products. It also uses transgenic animals to produce products for therapeutic and diagnostic purposes. and it involves search for cure of cystic fibrosis, cancer, and blood diseases. Okay, also, there is a term called xenotransplantation. This is the use of animal organs instead of human organs in transplant patients. Example is to genetically modify pigs to generate human organs so that chances of incompatibility and rejection is eliminated. So, have you heard of this term before, xenotransplantation? Let's look at this picture. So this is a xenograph platform. So you can see here we have our pig, and the pig cells are taken from the sample. And then we have the nuclear transfer, the cloning of these cells to create our genetically engineered pig. Okay, so from this genetically engineered pig, we have different xenoproducts such as organs, device tissues, and cells. So you might also wonder why does xenotransplantation use pigs as vectors? Because pigs actually have the same sizes of organs uh, to humans. It's comparable. Okay, so pigs are a good medium. So yeah, through genetic engineering, okay, here, especially in this part, so it's not directly uh, using pigs, raising them for harvesting their organs. Rather, cells are taken from different pigs and then cloning, of course, to create our genetically engineered pig. So that is xenotransplantation. So this is done to eliminate again the chances of rejection from transplant patients. So if you know, sometimes people who need a transplant, they find it hard to find donors. So maybe this xenotransplantation will reduce waiting time for patients. Okay, next, not only in the medical field, genetic engineering also has applications in agriculture. So I'm sure you've seen some of this from the video that I showed you earlier. So in agriculture, genetic engineering improves the crop plants. There are also animals producing human milk, if you remember the cows. And animals growing rapidly, herbicide and pesticide resistant crops. So I have here some examples from the pictures. This one here, this is called the golden rice. And then we have the flavor saver tomato, which is considered as a longer lasting tomato. And we also have our salmon, which grows into a really big size. And then we have our BT corn, or the Bacillus thuringiensis, which is a pesticide resistant crop. Okay, so genetic engineering also enables plants to have more nutritional values, manipulating to solve nutritional deficiencies. In an example, the third world countries, there is a vitamin A deficiency. So to solve this, people have been able to produce golden rice. Let's go back to the picture. This one over here, golden rice. This is golden rice. And this is a variety that contains beta carotene. That's why it's not the same creamy color or white color of the rice that we're used to seeing, because it is a precursor to vitamin A. It involves inserting a gene that produces beta carotene from a daffodil flower to the rice DNA. So the goal of producing the golden rice is to solve third world problems, okay, which is vitamin A deficiency. Right. Next, so those are just some applications that I showed you in the medical field and in agriculture. So, but there are also potential risks. Okay, so I'll show you some. First, they are potentially harmful to other organisms. like the larvae of monarch butterflies, which are not pests. As you can see, they are just butterflies. And they are harmed by the pollens of BT corn. So this causes them to suffer and die. Also, they elicit resistance. The exposure of weeds from GM crops could lead to the growth of super weeds, such as this weed right here that the man is holding. And it can also affect gene flow, since basically in genetic engineering, the gene flow is manipulated. The introduction of the genes from GM crops or genetically modified crops to nearby wild relatives develops new hybrids. So it really poses a challenge. Okay, if you think of it, how far can it go? Because it looks like a chain reaction is happening. Okay, so these are some potential risks that genetically modified organisms or food products pose. Okay, so I hope you were able to think about it, and I would like to hear your thoughts on it as well. But anyway, let us just refresh our memory with the things that we talked about. So, what are the steps in making a GMO? Let's see, don't forget. The first is to obtain the desired fragment of DNA from a source organism, and then insert the DNA into a plasmid, construct a recombinant plasmid, and lastly, insert the recombinant plasmid into a recipient cell. All right. So I think that's all for this video lecture. I hope you learned something from this. And if there are parts that are confusing, you can always go back. And if you want to view the first video lecture, just make sure to follow and view the playlist on this topic. Again, this is Ma'am Florence. Thank you very much, and I'll see you on the next video lecture. Thank you. Bye.